Excellent. It's my great pleasure to welcome you to the Daniel I.C. Wang Frontiers of Biotechnology lecture series. This is the second of this annual lecture series. And to begin, I'd like to start with a brief description of Daniel Wang, who is one of our highly esteemed institute professors and a member of the Department of Chemical Engineering. Professor Daniel Wang received the bachelor's degree in 1959 and the master's degree in 1961 from MIT in the Department of Chemical Engineering. And he went on to get his Ph.D. in ChemE's from the University of Pennsylvania back in 1963. He then spent 2 years in the U.S. Army Biological Labs at Fort Detrick in Maryland from 1963 to 1965, and he joined the MIT faculty in 1965. And has been a member of our faculty ever since. During the time that he was here, he founded the Biotechnology Process Engineering Center, which is an NSF Engineering Research Center initiative, and he was its director from 1985 to 1998. He also had a summer short course, which was the longest running and most successful short course series on biotechnology. Danny Wang was one of the true pioneers in biochemical engineering and biotech, developing some of its first texts, its first courses, and some of the first and earliest developments in this area. And here at MIT, he helped to launch a number of different initiatives and programs which helped to support that field. In 1996, Danny Wang became an institute professor, which is the highest honor that we can give here at MIT to our faculty. And since then, he has belonged to all of us, all departments. In 2000, he was awarded the Temasek Professorship at the National University of Singapore, where he devotes most of his time helping Singapore in their biomedical science research and development. Danny Wang has received many awards, including some from the American Chemical Society, the Marvin Johnson Award, the David Perelman Memorial Lecturer. He's also received several awards from the American Institute of Chemical Engineers, including the Food, Pharmaceutical, and Bioengineering Award, Institute Lecturer, and one of our highest honors, the William H. Walker Award in Chemical Engineering. He was given the Amgen Award in Biochemical Engineering from the Engineering Foundation, and he also received the Asia Pacific Biochemical Engineering Award. Professor Wang is a member of the American Academy of Arts and Sciences, the National Academy of Engineering, and the American Institute of Biological and Medical Engineering. Academia Sinica, which is from the Republic of China, and the International Institute of Biotechnology. So he has reached across the globe with respect to his impact. He's also reached across the globe with respect to the companies that he's influenced. He's consulted for over 50 companies worldwide and is on the board of directors and scientific advisory boards of a number of public and private companies. He's also authored and co-authored 5 books, over 230 publications, and 15 patents. He has a huge legacy. I'd also like to introduce the sponsor of the DIC Wang Lecture. This is our own alumni, Nubar Afeyan. And Dr. Afeyan is an MIT alum who is also a member of Danny's lab. So he has a very close connection both with the biotechnology field, our department, and with Danny. The sponsor of our lecture is the managing partner and CEO of Flagship Ventures, a leading early stage venture capital firm. Nubar also leads the firm's Venture Labs unit that invents and launches transformative startups. He's also a senior lecturer right here at MIT in the Sloan School of Management, working with our I-Teams course and several other courses on technology, entrepreneurship, innovation, and leadership since 2000. During his 25 career as an inventor, an entrepreneur, CEO, and venture capitalist, Dr. Afeyan has co-founded and helped build 26 successful life science and technology startups, many of them right here around us in Kendall Square. He was founder and CEO of Perceptive Biosystems, a leader in the bioinstrumentation field that grew to over 100 million in annual sales within 6 years from its first sales. Dr. Afeyan also received the Technology Pioneer Award in 2012 from the World Economic Forum in recognition of the breakthrough solar fuel technology being developed at Joule Unlimited. He currently serves as chairman of the Global Agenda Council on Emerging Technologies of the World Economic Forum. In 2008, he received the Ellis Island Medal of Honor. an award granted to outstanding Americans who have distinguished themselves as U.S. citizens and who have enabled their ancestry groups to maintain their identities while becoming integral parts of American life. Now, I'm very happy to introduce our speaker and guest for today, Dr. George Yancopoulos, MD and Ph.D. Dr. Yancopoulos is a native of Queens, New York, and he earned his MD and Ph.D. at Columbia University in biochemistry and molecular biophysics, finishing in 1987. He then accepted a junior faculty position at Columbia, studying the immune system. And shortly afterward, he won an 8-year grant worth well over the equivalent of $1 million in today's money from the Lucille P. Markey Charitable Trust, a foundation that specialized in promoting young biomedical researchers. Right around this point, Yancopoulos got a phone call from Leonard Schaefer, who was recruiting talent for a new biotechnology company. This startup would try to figure out ways to regrow nerve cells to potentially treat nervous system disorders, such as Alzheimer's or Parkinson's. Dr. Yancopoulos was intrigued. He decided to join the company, which became known as Regeneron in 1989, as its founding scientist. And he is currently president of Regeneron Laboratories and chief scientific officer. Over the years, he's helped direct the company's research into a variety of areas beyond the original nervous system scope, from eye diseases to cancer. Their first drug, Arcalyst, is a dimeric fusion protein consisting of ligand-binding domains related to the IL-1 receptor that was approved in 2008 by the US FDA for the treatment of an incredibly rare inflammatory condition known as cryopyrin-associated periodic syndrome. Dr. Yancopoulos was the 11th most highly cited scientist in the world in the 1990s. And in 2004, in 2004, he was elected to be a member of the National Academy of Sciences. Dr. Yancopoulos, together with key members of his team, is also the principal inventor and developer of Regeneron's other FDA-approved drugs, including Eylea, an injection, which is a treatment for wet age-related macular degeneration, or AMD. This affects at least an estimated 11 million Americans. And Zaltrap. Zaltrap is an anti-cancer therapeutic used in combination with chemotherapy drugs to treat resistant forms of metastatic colorectal cancer. Most recently, this July, Praluent, a PCSK9 inhibitor, was approved to control cholesterol levels. He's also developed foundation technologies, including the TRAP technology, Velocigene, which is a rapid, high-throughput method of gene modification, and VelociImmune for producing fully human monoclonal antibodies, and the basis of a novel humanized mouse capable of producing fully human antibodies. Dr. Yancopoulos is an extremely accomplished researcher, scientist, inventor, and entrepreneur, and we're very honored to have him here to give our second DIC Wang lecture. Thank you, Dr. Yancopoulos.
Thank you, Paula. That was quite the set of introductions. I have to say that I don't think anybody else did that much homework on me before. I learned a few things about myself. It's certainly a real honor and a privilege for me to be here today for many reasons, not the least of which is obviously MIT is a renowned hotbed of innovation and invention, something that is a treasure to the world and we all appreciate. Not to mention Danny Wang and the honor of giving this lecture, particularly being that he is such a leader in our industry in particular. So being asked to speak here is very special and meaningful. I also had the opportunity to just meet Nubar and spend about an hour with him. He may be the most fascinating man alive, I have to tell you guys. But being here is even more special to me because I have some old ties to this esteemed institute. And I'll never forget the last and first time before this that I was asked to speak at MIT. And it was in 1988, before I'm sure some of you guys in the audience were born, when I was 28. and it was by David Baltimore, then a famous Nobel Prize winner and the head of MIT's Whitehead Institute. And not only was David a hero of mine, like he was for many molecular biologists of my era, but as he put it when he introduced me for that talk, in which was another introduction that I'll never forget, he fondly considered himself my scientific grandfather. And that's because I had been the first graduate student of Fred Alt, who had been a postdoc with David here at the Whitehead Institute. And when Fred left David and MIT to start his own lab at Columbia, where I joined him as his first graduate student, in those early days, David Baltimore continued to work with and help take care of Fred and his graduate students, including me, and it was all one big family. And so that's how I was related to David and to MIT. And after I'd been with Fred for a long time and I was getting ready to leave was when David Baltimore invited me to give my talk here. But David didn't invite me to MIT just to give a talk. He was trying to save my career and maybe my life. You see, so first of all, I should give you just a little preamble about, because MIT is a very different place, and certainly the chemical engineering department is a very different place. And I'm sure, obviously, even back then, there were ties and links to industry. In fact, Danny Wang was pioneering. But in almost everywhere else in academia, even at your neighboring Whitehead Institute over here, industry was anathema. And you see, the reason I was up here and why David invited me was I had told Fred, my mentor, that I was going to be walking away from a faculty position at Columbia and leaving academia entirely, and instead joining this guy named Len Schleifer, that Paul already mentioned, to start a biotech company. And Fred panicked that it would be the biggest mistake of my life. And I mean, back then, in many worlds, and certainly in people doing basic biology research, biotech and industry were indeed dirty words, and going there was akin to going to the dark side. And so since Fred knew how much I looked up to and respected David, he asked David to intervene. So David decided to invite me up here, talk some sense into me, and he actually offered me a position here at the Whitehead at MIT. And I'll never forget the well-intentioned advice he gave to me when I was in his office. And when I walked out of his office, I can tell you I was shaking. And he told me that I had such a potentially promising career ahead of me in academic science, but if I did this biotech thing, I might never be heard from again. And those words echoed in my ears for years. And it made me second guess myself, and I long feared that maybe it turned out to be true, and maybe that's why, in part, I worked so hard to keep it from happening for real. So here we are, more than 27 years later, and I'm back at MIT almost full circle, and you know, it turns out that starting a biotech company wasn't such a bad thing, though it did take a really, really long time before that became entirely clear to a lot of people. And so the story of how we started the company and survived to tell about it is what I'm going to tell you guys about today. So I want to tell you about how we started this company and how we got to this point where we've done something that is actually very rare and maybe even unprecedented in our industry. And that is building one of the world's largest biopharma companies entirely from scratch. And remarkably, and like I said, perhaps in an unprecedented fashion, almost all of the key people who were involved in building the company are still here at our company doing it. And in fact, I think most of us feel like we're just getting started. So let me take a step back before I give you our story. And you know, you guys are one of the more sophisticated audiences probably about this topic, and I probably have less to tell you about this than most audiences, but just a little bit about biotechnology for those of you who might not be that knowledgeable. Biotech and biotechnology refers to medicines that are large molecules that are similar or identical to naturally occurring proteins and other complex biological substances in our body. As Danny Wang pioneered, these are generally too large and complex to make using chemistry alone, and they're produced via genetically modified microbes and cell lines. And it was the advent of those capabilities and genetic engineering that gave rise to the modern biotechnology industry that Danny was involved in helping sprout. And the one thing that most people don't appreciate, certainly I didn't appreciate, was how hard this business is. And I'm not sure I would have gone into it if I had appreciated back then how hard it actually was. It's incredibly difficult and time-consuming to develop a successful drug. And in fact, in terms of the reputation of our industry, right now our industry doesn't seem to have the greatest of reputations. But I think that it's very easy to defend what we're idealistically trying to do, which is one of the most important things that I think mankind can be doing, which is using science to try to fight disease and improve human health, and thus to make a difference in people's lives. So I believe, and I think it's easy to defend, that this is one of the most important things we can be doing as a society, but it's also one of the hardest things that human beings are challenged with. Trying to come up with a drug that impacts human disease and health is so hard that almost all the efforts result in failures. Estimates, depending on when you consider the start in the process, are more than 99% of efforts fail. On average, it takes 10 to 20 years and over $1 billion to successfully develop a single drug, and most efforts that are making these time and money expenditures actually fail. So the numbers are really daunting and humbling. Another way to look at it is there's over 5,000 biotech companies in the United States alone, and only about a dozen will ever produce an FDA-approved drug. And it almost never happens that a single company ever does it twice. In terms of big pharma companies, they have to a large degree given up on the discovery and front-end efforts and instead try to buy the opportunities from smaller companies. And in turn, the smaller companies often don't have the time and the resources to survive this incredibly long and arduous effort, nor the resources to get to it. So they concentrate on the early efforts and then ultimately sell out to the bigger companies. So from the beginning, we tried to build a different kind of company that, as I said, is representing something that I think is rare and rarer and maybe almost non-existent in this industry. That is a company that has the ability to go all the way from discovery through successful commercialization, and not once, but over and over again. So I think that one key thing that I want to say about how we did it is that I think the important thing is that we, and when I say we, not only Len Schleifer, our CEO, and myself, but a lot of the key early people who were involved, we all bought into the same concept, and I think that was key. And that was that we really believed that we could build a foundation in science and technology and innovation with the stated purpose of continuing to generate opportunities for the company, with the realization, the notion that it probably wasn't going to be the first opportunity or even the second or maybe even the 10th that was going to succeed. But eventually, if we really built such a foundation, we continue to innovate, eventually it would result in a major difference. And it's turned out to be that way. And now finally, after all this time, this foundation has led to what a lot of outside people and experts now consider one of the world's largest and most successful biopharma companies with an entirely homegrown set of technologies and pipelines. And one question that I get all the time is, how did we actually survive for all these years. As Len likes to say, a lot of people think that we became an overnight success after 20 years of failure. And there's, at least from certain perspectives, there's some truth in that. I always thought we were building something for all those 20 years, but from the outside, it certainly wasn't viewed as if we were a success. And I think it really just came down to this innovative spirit and foundation that we were trying to build and believed in. And it came down to all these key early scientists, almost all of whom are still with us today, who helped us really build the company and innovate. And what we kept doing was coming up with interesting opportunities that created apparent value that other companies, bigger companies, would want to invest in. And while we were so innovative on the science side, I think that we were also just as innovative on the business side. And I'm sure one guy who really appreciates that here is Chris Wiebacher, who's sitting here in the second row, who was the CEO of and Len's counterpart at Sanofi, and got to know my partner Len Schleifer very well. And Len really made incredibly innovative and I think mutually beneficial business deals, which reflected who we were and really made a difference in how we survived and who we became, which was that we made the first so-called 50-50 deals in the industry. The way it used to be before us and before Len had this idea was that small companies would come up with something that had potential and then eventually sell it, the idea, the product opportunity, whatever, to the bigger company and literally step out of the way, often disappear, just take their upfront money and be gone. Our attitude was we wanted to actually be involved in taking our children all the way through to adulthood. So we wanted to be part of that process, and we in fact wanted to drive that process when we felt there was nobody better than us who had invented or developed the story to take it all the way. And so what we wanted was a partner who was willing to largely fund that whole effort, but allow us to continue to play a big role, if not the biggest role in doing it, in exchange for also then getting a big stake of the profits, a 50-50 deal on the back end. And so this was really innovative. It was the first time that these sorts of deals were done. We'd like to think it's because the opportunities had such value, and in some cases we found some visionary folks on the other side who were willing to do these sorts of deals. Now, unfortunately, our first several of these deals did not work. That's why that bottom line on here is our stock price over our first 20 years. We would do something, it generate excitement, lead to a deal, the stock price would go up, the opportunity wouldn't work out, something would crash, would have a failure in the clinic, the stock price would go down, and basically we just bounced around between 0 and 20 or 30 bucks a share, so that's our stock price over the first 20 years. But the key thing was the whole time we thought we were building something on the inside, and we were learning how the next opportunity would have a greater chance of success. And that's how we survived. It was really about that. So this is just a series of developing innovative possibilities and opportunities, making deals, these 50-50 deals, carrying them out, and most of them not working out. And then eventually, one of them worked out. And I like to think that the timing was such so that once we figured out how to do it once, as you'll see, I think we really figured out how to do it over and over again. And in a unique way, where we still had that infrastructure that had built all this, was still intact, the people were there, they figured out how to do it once, and then that team, as I'll tell you, really learned how to just crank them out. But once we had our first big success, about 5 years ago now, as reflected in the stock price, this first 20 years was just noise in the stock price. And for those of you who follow such things, we have been the best performing stock on the S&P 500 over the last 5 years, with incredible rates of return, just showing what can happen when you actually hit it. And so all of a sudden it did seem like almost overnight we became a really big success. We're now one of the 5 largest biotech companies in the world. I think that there's reason to believe that we're the most innovative and productive company in biotech history. our pipeline, unlike any other in the industry, is entirely homegrown, from the basic discovery all the way through innovative technologies and breakthrough product opportunities that have been increasingly showing its productivity over the last 5 to 10 years. And we do it all sort of from beginning to end in terms of the process. And as I said, one of the reasons I think that we're so different is who we are, who the people are, and the fact that we're all still around and we're still building on what we've been trying to do for the last 27 years. And as I said, it's not only me and Len. Our board of directors, remarkably enough, once again, we're the only S&P 500 company where the majority of the board of directors are actually prominent scientists, starting with our chairman. For those of you who don't know Roy Vagelos, he's the father of the modern biopharmaceutical industry. You should look him up and read about him. He has been my hero since my father cut out an article from a Greek newspaper in 1975 and told me that I should, dedicate my life to trying to become like Roy Vagelos. And since that time, he's been my hero, and he still is, and remarkably enough, he's now the chairman of our company, and that's a whole nother story. But we have a lot of notable scientists on our board. We're not managed by businessmen. We're managed by scientists, and I'll get back to that as well. And the scientific management doesn't end, as I said, just at the top. We have the longest standing R&D senior scientific team in the business. Our key people, on average, have been with us for over 15 to 20 years. And in a business where it takes 10 to 15 years to do something, if you haven't been at a company for 10 to 15 years, you haven't done anything. If you go to every other company in our industry and you ask how long their people have been there, they'll tell you, on average, less than 5 years, which is why it's hard to imagine how they ever get anything done. And why? Why do our people stick with us? I think it's because of the unique type of company and culture that we've built in this spirit of innovation, where, once again, it's been increasingly recognized. Science Magazine has called us the top employer in the biopharm industry for the 1 or 2 for the last 5 years in a row, and Forbes calls us among the top 5 most innovative companies in the world across all industries. And I think what that leads, or what that reflects, is what we really feel on the inside. I have to tell you, I have the greatest job in the world, and if you ask, almost all of our scientists, they don't want to be anywhere else. Sorry, they don't want to be at MIT, they don't want to be in academia, they certainly don't want to be in any other company, because there's no other place that they can do more exciting science that can be translated to actually helping people than they can do at Regeneron. So that's why they stick around. And as I said, I think a lot of it came from a lot of our founding principles, our commitments to the science and technology. Particularly, we very much believed in genetics, and I'll get back to that as well as developing turnkey therapeutics approaches, and I'll talk to you a little bit more about that. But I wanted to give you just a few examples of how science led to new technologies, led to drug opportunities, built our foundation. And these are just some, of our homegrown Regeneron technologies. We've done so much, so many things that are incorporated into the normal scientific routine right now. I'm sure quite a few guys in the audience are like molecular biologist types, and you do protein engineering all the time. How many people here have done epitope tagging? Probably quite a few of you guys. Well, we invented that. So there's a lot of things that we've invented. But these are big things that have made a difference, and I just wanted to give you an example of how we started to build this collection of technologies that led to this machine that really does act as a wheel, where it starts on the left with the target discovery and the validation that's always been based on genetics. Initially, it was mouse genetics because it was hard to do human genetics 25 years ago. And initially, it was traditional mouse genetics. We improved on those technologies. We combined them with turnkey therapeutic technologies, which we're continuing to add on, added all the necessary downstream components from manufacturing, commercialization, and we really built this machine that's working, which we're continuing. The fundamentals stay the same. We believe in genetics. We're just now layering a lot more sophisticated mouse genetics, but also high-throughput human genetics in the process. A lot more types of turnkey therapeutics. And it really was a matter of building this collection of pieces that are highly integrated and work together, where all the people here are constantly communicating and interacting with all the people at every step of the process, which, believe it or not, is very unique in our industry. So just to tell you how we first built it and how it's almost obvious how it came right out of the science, into the technology, into the cures, into building this machine. Our first turnkey therapeutic technology is this technology that we refer to as traps. Now, we're probably more well known for our VelocImmune fully human antibodies. But what are our traps? Where did they come from? And how are they related to the genetics and so forth? Well, the traps represent a new therapeutic class that we invented at Regeneron, and it came from a very simple, basic science discovery that our scientists made, which is probably now in the textbooks. But as many of you I'm sure all know, the way a lot of growth factors and cytokines work is that they simply cross-link two different receptor units, okay? What's referred to now as heterodimerization. And in fact, most growth factors and cytokines and interleukins involve heterodimerization in their cell receptor activation process. Well, it turns out we discovered that. And I think one of these papers that I have listed here, I think maybe the second one, was the most highly, at the time that we discovered it was so important, this paper was the most highly cited paper in all of science in the year in which we published it. But our scientists didn't just make the fundamental discoveries, and particularly led by two of these fellows, three of them actually, Sam Davis, Neil Stahl, and Aris Economides. They then figured out, hey, we did the science, we discovered this, now let's take advantage of it. And they realized that if a normal receptor works this way, all they had to do was cut the ectodomains of this heterodimeric receptor off, so you only have the soluble components, fuse them to create a requisite heterodimer by fusing it to the constant region of an antibody. And now you would have something that mimicked the cell surface receptor, but because it was pre-formed, it actually would bind to its natural growth factor with a much higher affinity and much less likely to release it than the native receptor. So it would be a much better competitive blocker and could act solubly and could be actually a good pharmaceutical, a good drug, because based on the antibody properties that it was fused to, it would circulate around the body, sop up the growth factor, and really act as a very good blocker and inhibitor. In fact, at the time, these turned out to be the highest affinity blockers compared to all other reagents going back now, 15 or 20 years, compared certainly to humanized or early human antibodies, though I think now, especially with the advent of technologies like VelocImmune, that's now changed. But back then, these were unprecedented agents in terms of their ability to block their growth factors. So the two of them that we turned into drugs was interleukin-1 and VEGF. And how do we pick those and how we turn them into drugs? Well, this was the technology that gave us the drugs. How do we decide what to block? Well, that came from the genetics, and it came from the human and the mouse genetics. And as I said, back then, this is now 15 or 20 years ago, this was all done the more old-fashioned way. So we were working with some leaders in human genetics back then. And for those of you who aren't old enough to know how it was done back then, mapping human genes, 15 or 20 years ago was an incredibly laborious process that involved things known as positional cloning and cosmids and things like that. And it took many years. And we worked with a couple of leaders in the field to help map a mutation that seemed to cause a human autoinflammatory disease. It's known as a cold-induced inflammatory syndrome, in which simply a change in temperature or a drop of a few degrees triggered a huge overreaction in the body, which made the individual very sick. So you know how your mom used to tell you, don't go outside in the cold, you'll catch a cold? Well, these people, it really is truth, and it's almost instantaneous. They would get fevers, joint swelling and pain, aches, rashes all over their body, severe flu-like symptoms. And so working with these academic experts, we helped clone the gene to realize that this seemed to be due to unregulated release of interleukin-1. all over the body that was resulting in all of these phenotypes. So the first thing, and it's still true of human genetics today, you have a mutation, you think it causes a disease. How do you really prove it? Well, frankly, one of the best ways to prove it is to actually put that genetic mutation into a mouse and see if you can reproduce that disease in a mouse. And that's something that we believed in. If we could do that, then we knew for sure that we had nailed the gene. And not only that, so we did do that, so we created a genetically humanized mouse model of this disease. And moreover, not only did it reproduce the disease, but it gave us a mouse model to test therapeutics. And since we thought that the mutation was resulting in release of IL-1, we made an IL-1 trap. And we gave it to the mice, we cured the mouse, and we said, wow, this combination of human and mouse genetics coupled with this trap was a great formula, and it gave us great confidence going forward. We may have great success in a human clinical trial. Most things fail in clinical trials, but if you do them this way, it turns out the vast majority of what you do actually succeeds. So you turn the numbers around. Instead of 1 in 100 succeeding, it turns out that it's almost flipped so that, 90 or 95 out of 100 succeed. So these are the results from our first successful phase 3 trial in this study. And this is actually showing you the actual data. This is a very rare disease, so there's only 50 patients per group. And the key reason I show this to you is because most drugs don't work this way. Most drugs, including new drugs, they represent incremental benefit. They make the patients just a little bit better. Okay, rarely do they actually cure the patient. But when you do things this way, you get really impressive results. So this is on the left. This is the sort of key symptom score. It's referred to as the KSS for these patients. And these are the people on placebo, and their symptoms continue unabated. The symptoms, as soon as you give them the IL-1 trap for the treated patients, go down to essentially baseline. I mean, you've essentially completely cured these people. And then, of course, you do the same thing to the placebo patients when you treat them. So an 80 to 90% reduction in symptoms, this is not how drugs work. For example, the TNF drugs in rheumatoid arthritis, a very important big drug class, about 60% of the people have 20% improvement, and that's considered a big drug. Here, on average, you're getting an 80 to 90% improvement. And the thing that was most gratifying of all was getting the responses from the patients. And, for those of us, and I think many of you guys are probably, motivated in the same way, I was always fascinated by science, and I dreamed of using science to cure disease and make a difference in people's lives. There's nothing more gratifying or humbling than seeing how horrible these people's lives were because of their disease, and then what a difference you could actually make to these folks. And I'll just tell you briefly, like a story of, paying it forward. So one of the women in this trial was a mother. This is a dominantly inherited disease. It turns out 2 of her children were also affected. They were too young to be in our clinical trial, but when her life was changed with our drug, she applied to us for us to get compassionate use for her children. We did it, and both her children responded beautifully, and it changed their lives. They went from not being able to go to school half the time and not doing well in school, not being able to go outside and play, all these sorts of things, to being totally normal. And I'm going to choke up over here, but I get a handwritten letter in the mail. Back then there was actually mail. People would write them out, with a pen. This is probably about 10 years ago now or more. And it was the son of this woman. And he said, well, I looked you up in your history, and I saw that you got started in science by doing a high school science project, working at a company. And I saw what a difference science can do in my life. I've decided that I want to dedicate my life to coming up with the next set of drugs, and I want to get started by coming to work for you at your company so I could learn, how to do it. And that was 10 years ago. The kid is now still as dedicated. He's an MD, Ph.D., and doing phenomenally. And I think that, it is the example of paying it forward, because hopefully he will be making a difference. Anyway, so we were slapping ourselves on the back. We were so proud of this. We thought that we had really delivered to patients and so forth. and we got our first drug approval in 2008. I mean, the data came out a couple years before that because it takes a couple years to get your approval and all that, but we get it approved. So what did the world think of our great accomplishment? Well, probably one of the few times in history that a biotech company has a drug approved and its stock goes down tenfold. And all the pundits and the analysts said, you know, this is exactly, you know, why you don't put a couple of scientists in charge of a company. They're the stupidest, they literally called us the stupidest management team in the business. Why? Because we had spent over 10 years and hundreds of millions of dollars to get a drug approved that only treated 300 people in the United States. It was a great drug, but we were never going to make our money back, and we were refusing to charge $1 million or more per patient. So we weren't going to be making all that much money. I guess if we had charged $1 million per patient and made $300 million a year, it would have looked pretty good. But we were only making about $20 million a year. So, as I said, we didn't get much external recognition, and people just thought this was just further validation of why, you know, we were a lost company with no understanding of how to do things in this business. But we knew, or at least we hoped, that we had started to figure it out, and that we had started to build this machine that really was the foundation of everything that we still do today, and that all we had to do was continue to innovate, add to the machine, add more opportunities, and one of the opportunities would translate into financial success, and then would be viewed differently from the outside. And indeed, you know, that happened not that long after. In 2011, we had our second trap approved, VEGF trap, known as Eylea, and it became the third largest blockbuster in biotech history. And it really came about pretty much using the same exact formula that I showed for the IL-1 trap. So I'm not going to show you how we came up with it and so forth, but I just want to tell you a little bit about, you know, how impressively it actually can work in human beings. As I said, it's based on the same trap technology and the same sort of approaches that we used for the first trap, which wasn't such a big financial success. But it happens to work for the major causes of vision disturbing diseases in the United States. And so it really can make a lot of difference for a lot of people. And as I'll show you, a recent independent study by the NIH proved that it was the best in class, that it was, you know, substantially better than, for example, the antibodies that also attack the same target but were developed with the technology that was available 10 to 15 years ago when all these programs got started. So just to tell you guys a little bit about why the eye has vision loss in so many of the diseases, what's the common factor, and why it's all due to vascular endothelial growth factor, and why a VEGF trap that can block VEGF can be so powerful in treating all of these diseases and what they all have in common. Well, it turns out that, as you all know, this is a cross-section of the eye, this is the back of the eye, the retina. The retina is where you perceive light that is focused via your lens. And in fact, there is a single small spot in the back of the eye, which is known as the macula or the fovea. So this is actually an incredible technology that we didn't invent, but we use, that's known as optical coherence tomography, that allows you to visualize the living, breathing retina. And this very thin part here is the fovea, is where you have your best acuity. This is where what you need to be able to read, to drive, to check out your smartphone, whatever, right here. It's also it's the most metabolically active part of the body because it's constantly inputting information and processing it and transferring back to the rest of the brain. There's an incredible workload going on back here. So there's an incredible vascular blood supply to feed all that oxygen and so forth that you need for all this work that's being done right over here. What does that mean? That when something starts going wrong with your blood vessels, this is the part of the eye and the part of the body that first can be affected and can lead to all sorts of anatomic alterations. And since this has to be such a thin, perfect structure to correctly visualize light, as you might imagine, any anatomic perturbation right here can totally perturb the way you can see. And this is just an example of it. So basically, the vasculature that feeds in here, if it's not working well and it can't meet the oxygen demands, it starts growing out of control. And what you see here is you're literally causing a vascular inflammatory lesion right where you can least afford to, right where you need to have your thinnest retina to see. So all these little black areas here are either new blood vessels or leaking blood vessels that totally distort the anatomy, and you can go very rapidly from having 20/20 vision to best corrected vision. That means with the strongest glasses that you can put on, to being legally blind. So this is the same eye that I just showed you 2 weeks after a single injection of VEGF trap. This is, by the way, the number of letters on an eye chart. This is a special kind of eye chart that they use. Normal vision is about 75 to 80 letters. So these people actually qualify as legally blind because they can't read normal print, they can't drive, they can't do most of the normal activities of daily life. This is how thick their retinas are. With a single injection, you restore the normal thin part of the anatomy here, and their vision becomes almost normal, and a single injection is lasting for months. And as I said, there are 2 antibodies that were produced by Genentech that also attack VEGF and had been used in this setting. And luckily for us, the NIH was very interested in comparing all 3 of these because all 3 of these were being used at relatively high levels in these populations. That is, age-related macular degeneration and diabetic eye diseases, as well as an assortment of other eye diseases that result from vasculature problems in the back of the eye. And so, and this is something that's rarely done in the industry, a head-to-head study showing the vision gains. So there's 5 letters per line on an eye chart, and as you can see, we're 2 to 3 lines better than these 2 antibodies. So this is a really big difference because this can make all the difference in the world. Getting a couple of extra lines can be the difference between being able to drive or not drive, being able to read normal print, not being able to read normal print. So this got a huge amount of attention and ultimately led to us being the market leader in this area and contributed to this being such a big blockbuster, which it is. And of course, as I said before, it's the thing that's most gratifying about it are the individual stories, getting to see and meet the people and the patients and their families and seeing, you know, how science and what you're doing can make such a huge difference. And not only for me, for all of our people and the people who work on these projects, there's nothing more gratifying than to hear, you know, these stories about how what you've done has changed their lives and made such an impact. So that was my attempt to sort of give you guys just a view of how we sort of do things from the really big picture, using the genetics to help us define the targets and the pathways that we want to attack, and using a technology like initially the TRAPS technology to address those targets. We put them together. We are, unlike every other company in the world, initially agnostic as to commercial opportunity and size, but rather we go after the biology, and if the biology is there, we will follow up and we will make a drug for it, whether it's addressing 10 people or 10 million people. But just to also show you how we continue to innovate and develop, I just want to focus on a couple of our other key technologies that we've developed and just show how they have progressed over time. So initially, as I said, and in the first example, we were relying on traditional human genetics coupled to traditional mouse genetics. And then we started adopting or investing a lot in, we recognized the power of mouse genetics, and that there's frankly nothing, I don't care what you're doing with stem cells and iPS cells and human this or that, there's nothing more powerful than modeling the human disease by putting in the human genetic mutation into a mouse and having an animal reproduce a human disease. That gives you enormous confidence that you have the right mutation. It gives you a model to work in. And there's no other model system in vitro or anywhere else that can compete with that. And that's why, and I have to say, I recognized this a long time ago, which is why I had on our slides when we were first selling the company, this quote: "The company that best controls the genome of the most important laboratory animal model, the mouse, will become a leader in the biotech industry." So I'm very happy that that turned out to be true, because we invested a huge amount in building mouse technologies. And now our investment continues to deliver more and more sophistication in what we can do. We have the ability to put in mega base, that's millions of base pairs at a time, into a mouse, precisely replacing the mouse genes. But we can also do very subtle things like putting in human point mutations in inducible fashions into a mouse, so that until you turn the switch on, the point mutation doesn't turn on. So I just want to give you an example of how we've gone from old-fashioned mouse genetics. I should have pointed out we were the first company in its own labs to actually make a traditional mouse knockout, but now to what we're doing is really almost science fiction compared to what we could have even imagined 20 years ago. So just to show you the sophistication and the type of modeling that we do now, and the other thing about it is we can make these models incredibly fast and at very high throughput. So we can knock them out and produce a humanized model in a matter of months, and we can do hundreds, if not thousands, per year. So this is an example of another very rare disease in need of a therapy, and it's a very terrible disease. And our strategy, of course, is, unless we can model the disease in a mouse and really understand it, we're not gonna try to attack it. So in this particular case here, this is a disease where any sort of soft tissue injury results in a dramatic overresponse in the body, where you get a big inflammatory lesion or big swelling, but the worst part of it is the swelling almost overnight turns into bone. And these poor kids who have this disease, they can go to sleep and their neck is a little sore, and they go to sleep like this and they wake up and their neck is frozen in that position. Or they get a bump on their elbow, they bump their elbow, you get bone all over their elbow, and then they can't move their arm or their shoulder. And the worst thing is, with time, their entire rib cage gets encased in bone, and horrifically, they asphyxiate. So actually, one of our key guys who was involved in developing the IL-1 trap 20 years ago just published this paper on this new genetic model, Aris economedes, for this disease, where essentially he was working with our collaborator from the University of Pennsylvania, who mapped the putative gene mutation. The problem is, when you made the gene mutation constitutive, a lot of these are somatic mutations, the animals were born dead of other problems. So you had to figure out a way to essentially either create it somatically or turn it on at will. So basically, you can see, until he turns it on, the skeleton of the mouse is normal. As soon as he turns it on, they start forming globs of bone where bone shouldn't exist. So this is the lower of the leg, the knee, this is the thigh bone and the leg bone, and there's not supposed to be bone in here. You can see at high power. Eventually, the bone is not all connected, but just like in the human disease, all the bones eventually interconnect, essentially paralyzing and fixing the skeleton in place. So the first thing that Aris did was he modeled this, but then once he had a model, he was able to really study what was going on, and he came up with some amazing insights, and not only amazing insights, but a potential cure. So basically what he realized was something that's never really been seen before, that the mutation actually results in a gain of function in which a BMP receptor gains responsiveness to a ligand, which previously caused inflammation via a different receptor but could not form bone. But now this BMP receptor is mutated, so it now recognizes this ligand as an agonist. It was previously an antagonist, and instead of being controlled by this ligand, it actually forms bone abnormally. And once we realized this, very quickly, we were able to use this technology that I'm about to tell you in a second, VelocImmune, genetically humanized immune system in a mouse, to make an antibody against this ligand, which actually we've already given to the mice. It cures the mouse, and we will hopefully be clinical testing this within the year in the human patients who need this. This just shows how rapidly we now go, how we've compressed the timelines, but we're using the same fundamental approaches. We're just greatly amplifying and speeding up the technology. So it's just like we talked about for that IL-1 trap story that took us over 10 years. We do the same exact thing, just much more sophisticated, rapid fashion. We create the animal model, We cure it, we come up with a turnkey approach, this velocity technology, in this case, to create an antibody. Why did we switch to antibodies instead of traps? Well, because we were able to make the most amazing genetically humanized immune system mouse in which 6 megabases of immune genes were actually put into their precise locations in the mouse to create, by many measures, the most valuable mouse ever created. This is one of the things that Forbes cited when they highlighted us as one of the most innovative companies in the world. And once again, I just have to point out that there was a guy when he was a graduate student in 1985 that first proposed the possibility of making this mouse with my mentor, Fred Ault, who I hope is joining us for dinner, by the way. I invited him. But anyway, in any case, one could imagine, for example, engineering a mouse to make specific human antibodies. Such an antibody might even be used to optimize such an antibody, blah, blah, blah. Although conceptually outlandish, such genetic programming experiments may actually be realized in the not-too-distant future. Well, you know, I guess whatever it's been, 30 years is not too distant future. But it really became our new turnkey technology. When we were doing traps, there was no human antibody technology that could really compete with the traps. But this technology really does, and in most cases, we always still make traps and antibodies side by side, but in most cases, this new standard for making fully human antibodies creates the most powerful reagents. So, like I said, it all depended on this machine that we started building, and it all depends on the same key areas: genetics, turnkey therapeutics, and then incorporating clinical testing, manufacturing, commercialization, all the downstream components. And all we're doing is we're getting more and more sophisticated. I mean, biology doesn't change. Biology depends on genes and genetics. Okay, so we're never going to get away from that. And all our business depends upon is, as I noted 25 years ago or 27 years ago, is doing genetics better and better and better. And now we're doing mouse genetics, we think, as well as anybody, but we're recently getting into high throughput human genetics. Initially, we started with the traps. We incorporated these VelocImmune fully human antibodies that came from this genetically humanized immune mouse. And now all sorts of additional reagents we're now generating as turnkey therapeutics that maybe I'll briefly touch upon right at the end. And all this allows us now, at a much higher rate, to produce clinical candidates and validated clinical candidates in the sense that almost each one of these things has worked in the genetically humanized mouse model that gives us great confidence going forward, which is why our success rate going forward from going to the clinic phase 1 to phase 3 is exceedingly high. And as Chris probably remembers, when we made the Sanofi deal, everybody on both sides, but particularly on the Sanofi side, they were like modeling the success rate and how many things would fall off and so forth, and how much investment they would make. And I remember saying, you know, what nobody's anticipating is that we can't model the old success rates, because pretty soon we're going to have such a pipeline full of downstream things that are all going to need to be funded, because they're not going to be dropping off. And it turned out to be true, because almost invariably everything that starts out, when it's validated this way, does make it to the finish line. And so this has led to a pipeline that a lot of people on the outside have labeled as one of the most productive and exciting in the industry. And the amazing thing, it's all entirely homegrown. No other company can make that claim. So I just wanted to highlight two of our either recently approved or hopefully soon to be approved VelocImmune-derived antibodies that really, we think, once again, show the process. It's the same process. We're using, you know, we're not that imaginative. It's all the same approaches every time. But also what an impact that they might actually have on patients, which is what it's all about. So this antibody, alirocumab, is its, I don't know, you guys probably, maybe you know more about this than I do, but there's the scientific name, then there's the sort of generic name, and then there's the brand name. So this is a PCSK9 blocking antibody. That's what I would call the scientific name. Alirocumab, and there's a meaning for the U means humanized, monoclonal antibody. That's the generic name. And then the branded name is Praluent. But anyway, it's designed to hopefully reduce cholesterol and also lower cardiovascular risk. As I'm sure many of you know, the statins are one of the most important drug classes ever invented. They happen to have been invented by my role model and hero, Roy Vagelos, when he was head of research at Merck. And they probably had, other than antibiotics, as a class, they've had the biggest impact on saving human lives than any other pharmaceutical, probably. But despite the statins, there are so many people still at risk, especially based on changes in Western diet and exercise patterns and so forth. There's so many more people who are at risk who don't get to their target goals on statins. So despite statins, as you can see on the bottom line, there's tens of millions of Americans who don't get to a low enough LDL to avoid a high risk of a heart attack. So as I said, we were one of the first companies to go from, other than deficiency diseases, such as your neighbors at Genzyme focus on, in terms of genetic mutations that cause a disease, non-deficiency disease. We were one of the first to go from such a disease to a cure with the IL-1 trap. Well, this PCSK9 story is now replaced that first story as now everybody's favorite story of going from a gene to a potential drug that can make a difference. And this is a great story that started in academia. It started with, most notably with our collaborators at the University of Texas Southwestern, Jonathan Cohen and Helen Hobbs, who realized that there's a PCSK9 naturally occurring loss of function mutation in humans that's associated with low cholesterol and substantially decreased heart attack risk. And this led us and some others to first model this mutation in mice. So we're just doing the same thing we've always done. We start out with the human mutation, we model in a mouse, we confirm the story in the mutation to make sure that it's the right story. And then we use VelocImmune to create an antibody to it, and together with our collaborators at Sanofi, we were the first to treat human patients with this antibody. We were able to show that it lowered cholesterol levels, produced phase 3 data, and we were the first in the United States to get this approved, though Amgen has been with us essentially every step of the way. So there's now 2 PCSK9 antibodies approved, one from our labs and one from Amgen. And this just shows you the initial data that came out of the Helen Hobbs lab that got everybody so excited about it, which was, this is the normal distribution of cholesterol in the population. You see the average LDL cholesterol is about 150. These are the people who have PCSK9 mutations. As you can see, they're substantially shifted to the left. Their cholesterol is about 28% lower on average, and that 28% lowering of cholesterol over the entire lifetime leads to an 88% reduction in the risk of a heart attack. I mean, that's astounding. I mean, basically these people they're X-men, okay, and X-women. I mean, they have a mutation that makes them resistant to heart attacks. And so we and others got very excited about this, as I said, most notably us, closely followed by Amgen. We produced this antibody, and this is just the stunning initial human data, which just shows in a nice dose-dependent fashion. These are people on statins, on maximally tolerated statins who don't get to goal. With increasing doses of this antibody, you can get up to over a 70% lowering of cholesterol, getting almost everybody, almost every person can get to goal based on this antibody. And this led to a lot of excitement, a lot of publications and so forth, a very large joint phase 3 program with our partners at Sanofi. That involved over 20,000 patients and ultimately led to the approval. And the important thing is to study the effects on cardiovascular risk, you have to do these very, very large studies over time to show that you're substantially reducing heart attack risk. But the very exciting thing, and this was also seen with the Amgen antibody, that if you just looked at the smaller studies, even the smaller studies were showing in short-term studies on humans, I don't know if you can see it from the back, but you're almost halving the risk of heart attacks in these people even after short-term treatment with this antibody. Now, these are preliminary post-hoc data, which got us all very excited. The real results are pending this 20,000 plus patient outcome study. But this is really at this stage providing very exciting data that not only are we collectively with either our antibody or the Amgen antibody substantially lowering cholesterol levels, but that we could be substantially affecting cardiovascular risk, and the details were published in the New England Journal of Medicine. So this created enormous excitement and interest. The drugs were just approved this summer, and we're hoping that this story is going to really, bear out and really start making a difference in a lot of people's lives. And it really, hopefully, will become the showcase story for, the new showcase story for going from human genetics to important drugs that can really impact a lot of people. Once again, same story. This is now an antibody that is now hopefully nearing its last stages of testing in clinical trials, and the results are looking really impressive. A whole different classes of diseases. I'm sure you all appreciate that all sorts of allergic diseases seem to be tied together, whether it's standard allergies or whether it's skin rashes known as atopic dermatitis or eczema, or whether it's asthma. These are all seemingly related, and many people are trying to attack them with a variety of different mechanisms of actions. But what certainly the world recognizes is how these diseases, these allergic diseases, are dramatically increasing in frequency all over the world. It's been attributed to vaccines, or it's been attributed to too good sanitation, or it's been attributed to some environmental challenge. Nobody knows why allergic diseases are going up, but things that were non-existent 25 or 30 years ago, like peanut allergies, are now just commonplace. And just to show you, it does take a long time. About 15 years ago, we hypothesized that there were two particular interleukins that were the key initiating pathways across all of these allergic diseases. And we had a lot of genetic evidence. It was all done the same sort of way as I described. And we first made a trap to this. And the trap in very early studies worked, but this was before our other traps had been validated. So we didn't have an IL-1 trap or a VEGF trap that was approved at the time. And this was such a new technology, nobody was willing to invest in this thing. And so the program, due to lack of resources 15 years ago, died. Luckily for us, when we initiated our new collaboration with Sanofi that was built around antibodies, we said, well, we had this great trap. We were really interested in for allergic diseases. Nobody would invest in it. But now with Sanofi, we can work with them and invest in this exact target, but for allergic diseases. So we went back 15 years after having a trap that died and made an antibody to attack the same two pathways. And the results have really been stunning across, essentially, so far we've tested them in advanced studies across 3 different allergic diseases. So this notion of blocking these two cytokine pathways, IL-4 and IL-13, with this antibody seems to really work. So on the left side here, you have the atopic dermatitis data. The results are staggering. There's on average about a 75% improvement across the board in patients. So like I said, this is like what we saw with IL-1 trap in that rare disease. This is not like what you see with TNFs and rheumatoid arthritis, where the average response is a 75% response. And the same thing with asthma. We went in asthma in patients. First of all, we got in exacerbation. That is, that's the key clinical outcome that people are worried about. When you have severe asthma and you have to be hospitalized or visit an emergency room, that's called an exacerbation. We had, as you can see here, something on the order of 65 to 75% reductions in the exacerbations, but we saw dramatic improvements in lung function in people who were not well controlled on high doses of inhaled steroids, as well as what they call LABO, long-acting beta agonists. So this really seems like we have hit the fundamental pathway across an assortment of allergic diseases. And this just shows a patient actually sent us these photos because her life had changed and she just wanted to thank us. And she said, please, if you want to show these photos, use them, whatever. You guys have changed my life. This is what her skin looked like. I mean, her entire body was essentially covered with these rashes. They were incredibly itchy. She couldn't control herself from scratching. She was going to getting frequent ulcerations and infections. And her life was, as she puts it, just ruined. She was actually considering suicide. And within the first dose, she just entirely cleared up. And so there's been, so this is actually the note that she sent. And once again, I mean, this is what we live for, okay? That we've been working in the labs, in this case for more than 15 years, and we have a breakthrough, and it makes such a difference in people's lives. it's, as I said, that's what makes it all worthwhile. And the FDA seems to agree. They've designated breakthrough designation for this candidate, and so we're excited about this. But as I said, we have a whole pipeline behind it. Actually, I had lunch with the graduate students, and they were sort of saying, hey, they independently sort of said, wait, it looks like your technology is so rapid and quick compared to most others. Why don't you guys use it to address, emerging epidemics and threats like Ebola and MERS? Well, in fact, we have, and you guys can look it up. We've made some very nice relationships with the Defense Department and with BARDA and others to move our approaches for Ebola and MERS into testing. But the basic bottom line is that I want to get across is, what do? We had some very simple ideas about how to focus on certain key technological approaches and structure a foundation around that. And then once we started building it, to just add to those capabilities in a rational manner. So nothing's really changed. We start with the genetics. We're just getting much more sophisticated about it. We employ these turnkey therapeutic approaches, first traps, now velocimune antibodies, and I'll briefly touch upon bispecifics, or maybe not, incorporating all the other components. So, in fact, maybe I've spoken long enough. Maybe I'll just mention that in terms of our genetics approaches, we've just recently made a very big investment in human genetics, and we have one of the most ambitious human sequencing efforts going on in collaboration, in particular with Geisinger, which we think is a perfect partner, and I guess if anybody has questions, I can tell you guys all about that later. But it's certainly, in some ways, it's related to the President's Precision Medicine Initiative. It's just that we'll probably be done with this effort before they even get started. But I hope what I've gotten across to you is the story of our company and how we built it, why we think it's special, and why we're really excited about this foundation that we have, and how we think that, and we hope that it's going to continue to deliver important new medicines to people who really need them, and that we're going to be able to be doing this for a really long time going forward. So thanks for your attention, and if we have any time, I'd be glad to take any questions.
Yancopoulos. Before we start questions, I'd like to present you with our plaque, thanking you for serving as our lecturer. And here it is the faculty of the Department of Chemical Engineering at MIT presents this plaque to George D. Yancopoulos in recognition of his selection for the second annual Daniel I.C. Wang Lecture on the Frontiers of Biotechnology. Thank you very much.
Thank you, Paul.
And you can mail this to you. We have time for questions. I'm sure there are questions in the audience. And I think we have a microphone that is traveling. A mic volunteer? Yes. All right. We have microphones on either side. You can come up too.
First of all, outstanding story, outstanding successes. Thanks for sharing, especially the personal component is very, very touching. So my question is, there's 1,500 genetic regions that are reliably associated with disease. There are dozens of candidates that are out there. You basically showed a very short list of things that are sort of moving through, and I'm just curious, what is your rate limiting step? Basically, if you were to sort of improve upon this pipeline, what would it be? Would it be, I'm sort of curious, what's next? Is it the technology itself that you can see next generations of that technology coming through? Is it our understanding of human genetics that's holding you back in some of these cases of what is the mechanism of these regions? Is it simply the throughput of taking your existing predictions through this pipeline? So what would you improve if you could improve something?
These are all great and very pertinent questions. So the first thing that we always do is when there is a good candidate, we try to model that mutation. We knock that exact piece of DNA into a mouse and see if it reproduces a disease. That isn't that limiting for us because we can do hundreds, if not thousands of those per year. Okay? Success there, you know, if you do or you don't model the disease. It doesn't mean it's necessarily wrong if you can't model it. But being able to model it then gives you something that you can feel sure about and that you can then use as a model. The second real limitation is even if we even model diseases that are not likely to be targets for our types of therapeutics. But that's the next limiting thing. If we have a target and a pathway that, as you say, is well-defined and obvious that we can target it with a biologic, then we're off to the races. But probably the majority of the mutations, even if they can reproduce the disease, are not necessarily attackable by a biologic. And that brings up, I think, opens up enormous opportunities, you know, for the entire industry. We don't necessarily think that our approaches are going to solve every kind of genetic disease, but the ones that we can, we think that we're going to have a huge competitive advantage on. But there's enormous opportunities out there, which we're also trying to now get to. How do you attack, you know, disease settings where, you know, you have a genetic mutation, but you have to go back in and either correct the gene or replace the gene or do something intracellularly that you can't do with these biologics. So that's a great limitation for a lot of the genetic diseases that we're actually defining with our human genetics approach and that the world is defining. And I think that that's just a great opportunity for us, but also the whole field. There's a lot of other people with a lot of other approaches. So we think that in the biologic space, we like to think that we're leaders and we're going to compete well against anybody. In these other areas, you know, we're trying out there, like other people, trying to develop new approaches to really attack these new types of pathways.
My second question is that there's effect sizes differ in the genetics of different disorders. Basically, there's, you know, of course, rare mutations that travel in families. There's coding mutations that are common in the population. And then there's all the non-coding mutations that affect some regulatory circuit and so on and so forth. My question is, what's your take on, you know, the therapeutics? Is it really that you're just going to reverse the effect of the genetic mutation? Or are you looking at genetics beyond that? Are you looking at the genetics as sort of the things that you're trying to fix?
Yeah. Well, this I think is the next frontier, is in some cases, whether it was for that cold-induced autoinflammatory syndrome or for VEGF in the eye or for PCSK9 or for this allergic situation where you have a well-defined pathway, genetics, target, it all makes sense, and you can attack it from the outside. And I'm not sure that mankind can develop a better medicine than the naturally occurring antibodies that you can produce out of a mouse. But for a very large proportion of these genetic diseases and other types of diseases, these solutions aren't going to work. And you're going to have to get inside of a cell and figure it out. If you have a non-coding, unless the non-coding results in some pathway that you can define, I mean, many times you need the mouse model in order to define the pathway because you can't really do studies in human beings and, you know, to understand chemistry and biochemical pathways and take out biopsies constantly and so forth. But sometimes those pathways that you identify are not going to be active in A, which I showed you for that rare bone disease, where now, oh yeah, we have an antibody that can block it. If that mutation had led to some sort of intracellular activation of some new pathway that we couldn't target with a biologic, you need a whole new approach to doing it. So the majority, I think, of the genetic alterations that are linked with disease are probably not going to be amenable to biologics, which just opens up huge opportunities, I think. I think another part of your question was, I think a lot of the best genetic mutations for doing our approach and for modeling are ones that are highly penetrant, meaning almost Mendelian, meaning that, you know, they have a high percentage of showing the phenotype in man, which is the only way you're going to see the phenotype, you know, in a mouse. If all you're doing is increasing your odds of getting the disease by 10%, which is like a lot of these GWAS hits or so forth, at best you're getting a 10% increase in your risk of getting the disease. The odds that you're going to easily model that in a mouse are very, very low, unless you understand what the whole modifiers might be that be putting those other people at risk. So on the one hand, I mean, what we do is we go where the biology is most obvious. And one advantage of being able to do high throughput things is you pick the ones where your approaches are going to work. What I'm not showing you is when we model 100 or 200 mouse models, and 90% of them don't give an obvious phenotype, or if they do, they give a pathway that's not addressable by a biologic. But that's the advantage to numbers. If you have high enough numbers, you get hopefully at least a few a year that are going to make a big difference doing it this way.
Thank you.
Do we have additional questions?
So it was a great presentation, George. I have to say I still follow every quarterly result with a lot of satisfaction and pride.
For those of you who don't know, this is Chris V. Bacher, our CEO of our partner Sanofi. We were very saddened when he left Sanofi, but we're still partners in heart, right?
Absolutely. But, I can say I knew you when. And I can tell you in 2009, when I became CEO of Sanofi and I decided to invest more and expand our collaboration, there was nobody that was even thinking you'd ever show those slides. And in fact, you just finished a 20-year period of essentially providing absolutely zero shareholder value. And I think there are a number of...
some shareholders made money on the way up and some of them made on the way down, but on average, you're right, yes.
And I know a few of those. But there are a few probably budding entrepreneurs here. And I think what's really unique about Regeneron is that you could have the time, 20 years, really to pursue science. And I think there are a number of unique models because, in a world today where venture capitalists have a 5 to 7 year time horizon, public market investors have an even shorter time horizon. The fact that a single management team could go 20 years without really delivering the goods in terms of shareholder value, but delivering the goods from a scientific point of view, I think is really without parallel in our industry. I cannot really think of another company that's gone that long and then had your phenomenon. Because today it's obvious. Today you're worth north of $50 billion. But back then, it was different. You did some, I think, unique things with your share structure. You went and did an IPO early, so you weren't dependent on VCs. But maybe you could give some advice as to how can you get through, because we all know that science takes an awful long time. And one of the biggest problems we have is how do you match the time it takes to mature science with the very short time horizons of investors? And if you had any advice as to how you got through that period, I think it would be helpful to some of the entrepreneurs in the crowd.
Yeah, no, I think that these are great points. And I also think that, just to point out, I do think that Chris recognized that we were on the verge several years before anybody else did, because as you said, that was at a critical point in our history and in our relationship where you came on and you decided to, in the face of other people calling you crazy, increase, your investment. Essentially, the pipeline that we produce is due to that investment. But most people, certainly in 2009, weren't seeing this. But as you said, that's a critical thing, that in this business where it takes so long to do anything, the problem that we have is we have, in too many cases, and I think this is a problem for society as well as government agencies that are trying to, control healthcare costs while somehow promoting, science and innovation in the country. Everybody thinks in too short terms. And we know these timelines, unfortunately, we're not designing an app that's going to turn, people into billionaires overnight. We're doing things that just the human testing is going to take years. And there's no way that, it's not we're going to come up with great new technologies that are going to dramatically shorten it. Yes, we're going to be cutting off a year here or there. So the things that, were different about us was, I think that there was, as I said, not only Len and me, but a whole group of people who really bought into the long term. Two was that our deals were designed with that. As you said, most people have short-term horizons. And there's nothing wrong with it. People want to make their money, whether they're VCs or other people who want to get in and out. And we were luckily, and to Len's, enormous credit, because we had an early IPO, but because of the structure of these 50-50 deals where we got partners who were willing to fund our research, we didn't have to go to outside funding. Okay, that allowed us to continue to build and innovate and so forth. And if that failed, which is, as we know, most early things fail, we had the next thing that somebody else was willing to invest in. And that kept us going. As you also said, you know, there were some unique things in the share structure that, once again, Len actually innovated on, which are now, as you know, illegal. So Len very cleverly, I mean, I didn't know anything, so I thought that everybody did it this way. I didn't realize how brilliant Len was, but he gave the founders voting shares, had 10x voting rights. So what did that mean? That meant that we could sell 90% of the company and we still controlled the company. Also, when we made, and now, as I put it, that's considered, that is illegal, but to have these different classes of shares. But also, when we made our agreements, as you know, we were, and you recognized early on, I think you were actually quoted something to the point of, you know, you were interested in us, you didn't want to buy us because then you would sanitize us, you wanted to leave us who we were. So that was one of the key things, finding partners who recognized that and didn't just want to buy us for that one asset, but they wanted to keep us who we were so that we could continue to sort of produce things. And just in case a Chris Wiebacher would leave, you know, Len wanted to make sure, as you know, that we put in all these sort of standstills in our deals and so forth, so that the big company would have all these, we would have all these protection against the big company wanting to buy us. Because the thing, you know, as Chris realized, but as most, I have to say most people don't, they look at companies and like us, especially in our early days, as like a one asset opportunity. Oh, they have this one drug, we're gonna buy them for the one drug. We don't care about anything else. It's that one drug that might give us some revenues, okay? But Chris recognized that, hey, this could be, you know, a machine that could keep turning things out, and we don't wanna mess up that machine. But in many cases, companies have been taken out. They may have had big futures, but they were taken out by somebody who just wanted that one drug that company had, and then the company was gone. So it was a collection of all of these things. And some things, you know, we sort of joke about. Some of these things were well planned out and we thought about, but we have to also admit that, you know, there was a lot of luck involved. I mean, there were times where we could have gone out of business, you know, where, you know, for years, and I don't know why, it never used to bother me. It used to bother Len a lot. But, you know, for years we would go on and we only had 6 months of funding. That was the joke. We would have our quarterly meetings and we'd meet with our board of directors and, you know, we'd go through all these incredible financials. At the end of the day, Mike Brown on our board, Nobel Prize winner, would ask, so what does that mean? You have 6 months of funding left? And that was every board meeting. It's exactly what we had. But somehow I knew. I knew that, oh, Len would figure out a way to, you know, get the resources so we could sort of keep on going. But things just could happen. The markets, that could have been a terrorism attack. 9/11 could have happened at the wrong point. All these sorts of random events. So what you also have to recognize, I think, what I recognize in this business, that no matter how well you design things, no matter how well you plan, no matter how many protections you put in place and so forth, you know, a really, really important component is luck. The luck of the people who get involved. I think we were incredibly lucky to get the right group of people involved. We were incredibly lucky in sort of our advisors. We were incredibly lucky, at least in some of our collaborators and the big companies that we partner with, and our timing often benefited a lot from luck, where things could have gone very differently. But this is why, you know, as Len, my partner, you know, says a lot, you know, he doesn't think this is ever going to happen again because of the timelines it takes. Nobody's going to have the patience anymore, and you're not going to have this collection of things that miraculously happened to allow this all to occur. Part of it was design, part of it was planning, you know, but a lot of it was luck.
Well, I'm afraid we don't have any more time, but we had a wonderful time listening and learning from you, Dr. Yancopoulos.
Thank you.
Thank you very much.